Half of the 2019 paper.
Okay, second half of the 2019 paper, question 14 through. Okay, which statement describes an event that occurs during the lag phase of microbial growth? Okay, so you're expected to recognise 4 phases: lag, log, stationary, and death. Okay, the lag phase, the word you're looking for here is induced or induction of enzymes. Okay, we've then got our fastest possible growth going on here, so nothing is a problem. Then we have the stationary phase, and in stationary, the most likely thing that we're looking at here is about secondary metabolites, which are sometimes quite useful to us. And in the death phase, really what we're looking at here is the idea that we have a massive buildup of toxic waste products. We'll put at the end there so it doesn't sound like it needs to be quarantined off. Okay, so in the lag phase, we're looking for enzyme induction. And there's B. Okay, so this would have been the stationary. This would have been this would have been your exponential, or your log phase, if we prefer, and depleted of nutrients because we run out of everything. Could be actually a little bit of the stationary, but really more likely we're talking about death. Okay. Number 15, using recombinant DNA technology, a culture of the bacteria species Micrococcus luteus was genetically modified by the plasmid containing a gene for a human protein. Protein was synthesized, but the genetically modified bacteria failed to fold correctly. Which of the following changes to this procedure may lead to a correctly folded protein being produced? Okay, so what they're saying here is you managed to make it because it was synthesized, but it didn't fold. Okay, so what you need to recognize here is it's not a case of having a problem with it being produced. So it's not about putting a regulatory sequence in because it managed to synthesize it. And it's not about using an artificial chromosome instead of a plasmid because you managed to make it. The reason you might use an artificial chromosome is if you wanted to insert a larger quantity of DNA. Okay, so what we've got is use a different species of a bacteria or use yeast cells. The yeast cells is what you need to recognize. Okay, yeast cells are also eukaryotic cells. So if you've got a human protein, one of the problems with putting that in bacteria is bacteria do not do all of the kind of translational things that eukaryotic cells do. So yeast cells, however, which are eukaryotes, they will. Okay, so if it's to do with folding, you need a yeast cell. If it's to do with the size of the code, then it's an artificial chromosome. Okay, question 16. We've got a lovely little graph here. Okay, bacterial cells were exposed to disinfectant for 14 minutes. Every 2 minutes, a sample was taken, number of viable cells counted. The reason I'm saying lovely little graph is because what we have here is a log scale on the going up the way. Okay, so what you really have is this is 1, 2, 3, 4, and now I can't fit it in, but 5, 6, 7, 8, 9. You can see it's kind of squished up. And then this would be 20, 30, 40, 50, 60, 70, all the way up to 90 is a wee titchy thing in there. And then we're going, that's 200, so that would be 1,000 on the next one, and 20,000. Okay, hopefully we can see exactly where this is going because they really are expecting you to actually pull off everything perfectly. Okay, we're looking for the percentage decrease, right? So percentage decrease means we are looking for the change, okay, divided by the start, times it by 100. Okay, so the change here, right, so we're looking for 6 minutes. So 6 minutes is this point here. And this is going up. Oh, that doesn't read that should be 2,000. Okay, so as this goes up, that was 1,000 at the bottom, so that's 2,000, 3,000. This is 4,000. So to get the change, what I'm going to do is take 1,000,000, take away 4,000. Okay, to get my change. Divided by the start, which is my 1,000,000 again. Times that by 100, and we get 99.6%. Okay, large decrease. Okay. Question 17. Field trial was carried out to investigate the effect of applying different masses of phosphate fertilizer on grain yield of a wheat cultivar. The wheat was grown in 3 fields. Mass of 50, 100 or 150 kilograms of phosphate fertilizer per hectare was applied. How could the design of the field trial be improved to minimise the effect of bias? Okay, so we're looking for trying to remove any possibility of kind of systematic things being a problem. Okay, so repeating the trial using a wider range of masses of phosphate, that would change the trial, so it's not about that. Increase the number of replicates, that would help you maybe get a more reliable result, but that's got nothing to do with bias. Carry out another field trial exactly the same using a different wheat cultivar. That's changing the experiment again, because now we're looking at a different wheat. D, however, makes sense. Divide each field, apply randomly the different masses of phosphate fertilizer. It would mean that you have no bias about where they are, whether they're at the top of the field, the bottom of the field. Instead, they're all over in different spaces. So you might have your plot divided up, and randomly you might have, there's a 50, there's a 50, there's a 50, but here's a 100, and you just basically spread them out with preferably using kind of a computer generated sample to decide where everything goes. Okay, so it's completely randomized, and that hopefully gets rid of any bias that you might have. Okay, question 18. Fungi were cultured in a fermenter to produce an enzyme that was released into the culture medium. The graph shows the effect of oxygen concentration on the dry mass of the fungus and the concentration of enzyme in the medium after 48 hours growth. You should be starting to see this as quite a common thing. Okay, so we've got the dry mass of the fungus, and we've got the concentration of the reading this way. Okay, so dry mass of the fungus, look at this one here, is this one here. Okay. And we're looking for the concentration of the enzyme would be the open dot one, so that's there, and that's reading to this scale. This is all about reading the correct way. Concentration of enzyme when the dry mass of the fungus was 36 units per litre was what? Okay, right, so looking at the fungus, so I'm looking at the solid. Okay, I need 36, so this is going up in twos, okay, because there's 30. So 32, 34, 36. Read it along, find it, it's here. Okay, so that was at 2 oxygen units. I need to find out what the enzyme was, so let's read up to the enzyme line. Here's my sample, here's my number. Read it across this way. Okay, this one is going up in ones. Okay, so we are 28. And B. Okay. Breeding programs can be used to increase starch levels in barley plants. This involves breeding closely related plants with high starch levels for several generations. Okay, so you're trying to work on continually breeding back in higher starch levels. Okay, so what you're trying to do is to get rid of the differences. Okay, so you're trying to get everything pretty much the same. So what you're trying to do is homogenize the kind of the phenotype that you have. So what you're trying to do is reduce the number of heterozygotes that you have. Increasing frequency of heterozygotes would be completely the wrong thing to do, because what you then have is more and more differences, and we're trying to get them all pretty much the same. And then I really don't want to increase the frequency of homozygous deleterious alleles. That's inbreeding depression. Reducing the frequency of that might be something you have to do later, but not at the start. Okay. Question 20. Bar chart shows the percentage decrease in yield of 4 crops as a result of the effects of weed, fungi, and insect pests. Okay. Predict which crop will show the greatest increase in yield if herbicides and insecticides are applied. Okay, so herbicides, okay, kill weeds. So that's this bit here on that one, this bit here on this one, and this bit here on this one, and this bit here on this one. Okay, insecticides will kill the insects. Okay, so that's this bit here, this bit here, this bit here, and this bit here. Now all you need to do is just count them up. Okay, what we're looking for is which one would have the best change in their decrease in yield by getting rid of the weeds and the insects. Okay, and if you count them up, you'll see that this one has the biggest change. Okay. Oh, Sorry, I can't find them. Oh, there we go. Okay, the diagram shows the breeding of 2 paternal pig breeds to produce the modern Duroc pig. Okay, you've got the brown Duroc crossed with the Jersey Red gives you the modern Duroc. The modern Duroc pig has increased quality and quantity of meat compared to both parental breeds. The development of the modern pig breed is an example of, it's almost a perfect definition, to be honest, and one that you might want to use. Okay, we're looking for cross breeding. Okay. Question 22. A study into animal welfare in a zoo was carried out by observing the behaviour of some of the animals in their enclosures. Behaviours observed, not absorbed, are shown in the table. Which 2 animals show signs of poor welfare? Okay, so for poor welfare, we're looking for an excessive either manic kind of energy or a complete apathy. Okay, so energy kind of levels or visible things about energy being super high or super low. Poor reproductive behaviour, and stereotypy is your other kind of major one. Okay, so this sun bear you would consider feeding young as normal. Okay, we would consider that a subordinate monkey grooming a dominant male, that would be perfectly normal. Okay, repeatedly chewing bars of enclosure, not so good for the white rhino. And sleeping in the sun for a spotted hyena, we would say, yeah, okay, that's probably normal. Okay, the pacing in the lion is not normal behaviour either. Okay, so in terms of which ones show poor signs of poor welfare, it's the lion and the white rhino. Okay, which of the following is not a behaviour of a worker honeybee? Okay, so the worker honeybee, again, straight definition, doesn't produce eggs. They do, however, defend the hive, gather pollen, and they also feed the offspring. This would be your nice example of kin selection. Okay, question 24. The statements describe examples of behaviour used by animals as protection from predators. Which examples are social defence? So social defence means that you need to have several of your pals of the same group as you doing useful things. So a hedgehog rolling into a ball, that's an individual defence, that is not social. Mackerel swimming in a large shoal, so it's difficult for the predator to single out. Yep, that's a definitely social thing to do. And an individual meerkat watching for predators while the other ones feed, that's definitely social as well. They would obviously take it in turns. Okay, so see? And last question on the paper for this multiple choice. The list describes some aspects of biodiversity. Number and frequency of different alleles, the proportion of each species in an ecosystem, the number of different species in an ecosystem. Okay, and then we're looking for which are components of species diversity. Okay, that's pretty much, again, a straight definition here. This is genetic diversity we're looking at here. Okay, but these two are good for species diversity. How many different species you have, and then the proportion of each species that you have there.